Good morning, everyone. I'm delighted to welcome you all today. Thank you for being here. I'm excited to present to you the WHO Good Practices for Microbiology Laboratories, Part II. My name is Dr. S.K. Singh. Over the course of this session, we'll explore key guidelines and best practices to enhance laboratory quality and safety. We'll cover important aspects to ensure our microbiology labs operate efficiently and reliably. Let's get started. Today, I want to focus on the importance of reagents and culture media in microbiology testing. First, it's crucial that we use reagents and media of appropriate quality. High-quality materials ensure the accuracy and reliability of our test results. Next, we need to verify the suitability of each batch of reagents we plan to use. This is especially important for reagents that are critical for the test because any inconsistency can lead to false results. We should do this verification initially, when we first receive the batch, and also throughout its shelf life. Regular checks help us catch any degradation or contamination that might occur over time. By maintaining strict quality control of our reagents and media, we uphold the integrity of our microbiology practices and ensure our results are trustworthy. This is a key part of overall laboratory quality management and directly supports the accuracy of our diagnostic conclusions. Let's start by talking about the media we use. We purchase media exclusively from approved and qualified suppliers, ensuring quality and reliability. It's also important to note that we perform growth promotion tests on all media, and we do this for every batch and shipment. This step helps us confirm that the media will support the growth of the target organisms consistently. Now, aside from growth promotion, we conduct other critical tests on culture media, diluents, and suspension fluids. These tests are designed to verify several key properties. First, we assess the recovery or survival of target organisms, aiming for a recovery rate of 50 to 200%. This means that after inoculating with no more than 100 colony-forming units, we should be able to recover an appropriate number of organisms, indicating the media support their growth effectively. We also test for the inhibition or suppression of non-target organisms, which helps ensure that our media are selective enough for accurate identification. Additionally, biochemical properties, both differential and diagnostic, are checked to confirm that the media can help distinguish between different microbial species. Finally, we examine other important properties such as pH, volume, and sterility, which are fundamental to maintaining the integrity of our testing process. All of these steps are part of our broader microbiology laboratory practices, which are essential for ensuring the accuracy and reliability of our microbiological analyses. This thorough testing underpins our commitment to quality and precision in microbiology. Let's continue our exploration of microbiology laboratories with Part II. In this section, we'll delve into some of the more advanced aspects and specialized techniques used in these labs. It's important to understand that microbiology labs are not just about identifying bacteria. They play a critical role in diagnosing diseases, guiding treatment, and even in research for new therapies. We'll look at the different types of testing methods employed, such as culture techniques, staining procedures, and molecular diagnostics. These methods allow us to accurately detect and identify pathogens, which is crucial for effective patient care. Additionally, we'll discuss quality control measures that ensure the reliability and accuracy of laboratory results. Maintaining strict standards is essential because decisions based on lab results can significantly impact patient outcomes. Connecting this to the overall theme, understanding these advanced lab practices highlights the importance of microbiology in the broader context of healthcare and disease prevention. It's about combining technical expertise with rigorous standards to support clinical decision-making. Now, let's talk about preparing media properly. It's essential that all media are prepared in accordance with the manufacturer's instructions. This ensures that the media will perform as expected, providing accurate and reliable results. Another critical aspect is paying close attention to specifications, such as time and temperature, during sterilization. Proper sterilization is key to preventing contamination and ensuring the integrity of our experiments or diagnostics. Additionally, I want to highlight that microwave devices should not be used for melting media. Microwaves can cause uneven heating, which might compromise the media's sterility or other properties. Instead, we should use appropriate sterilization equipment to achieve uniform and safe results. All of these points tie back to our overall goal, maintaining the quality and reliability of our microbiology work. Proper media preparation and sterilization are foundational to accurate microbiological analysis. When it comes to cooling and repartition of media after sterilization, it's crucial to perform these steps under unidirectional airflow, or UDAF. This helps ensure that the media remains sterile and uncontaminated during handling. For plated media that will be irradiated, we might need to add specific additives like antioxidants or free radical scavengers. These substances help protect the media from the potentially damaging effects of the irradiation process, maintaining its integrity and effectiveness. After irradiation, it's important to validate the media. We do this by performing quantitative growth promotion testing on both irradiated and non-irradiated media. This step confirms that the media still supports microbial growth and meets quality standards after irradiation. All of these procedures are part of maintaining the integrity and reliability of microbiological media, which is a key component in ensuring accurate and reproducible testing results in our microbiology laboratories. Let's continue exploring microbiology laboratories with Part II. In this section, we'll delve deeper into the essential aspects of working in these labs. We'll discuss the key techniques used to identify and analyze microorganisms, which are crucial for accurate diagnosis and treatment. It's important to understand how these labs contribute to overall healthcare by providing precise and timely information. We'll also look at the importance of maintaining strict safety protocols to prevent contamination and ensure the safety of lab personnel. Remember, the accuracy of laboratory results directly impacts patient outcomes, so attention to detail and quality control are paramount. By connecting these points, we see how microbiology labs are integral to the broader healthcare system, supporting both diagnosis and research. Let's talk about labeling in the microbiology laboratory. It's really crucial that all reagents, including stock solutions, media, diluents, and any suspending fluids, are properly labeled. This helps ensure everyone knows exactly what they're working with and reduces the risk of errors. The labels should include key information such as the identity of the reagent, which tells us what it is. We also need to note the concentration, so we know the strength or potency of what's inside. Storage conditions are equally important, where and how the reagent should be stored to keep it stable and effective. Don't forget to include the preparation date and the validated expiry date, or the recommended storage period. This helps us track how long the reagent remains reliable. And finally, the label should clearly identify who prepared it, so there's accountability and a way to trace back if any issues arise. Overall, proper labeling is fundamental to maintaining quality and safety in our microbiology labs, ensuring that everything is traceable, reliable, and ready for accurate testing. Organism resuscitation is an important concept to understand because sometimes test methodologies can produce cells that are only injured but not dead. These are what we call subtly injured cells, and they can be tricky because they may not grow or be detected properly without some intervention. This often happens due to various stress factors like exposure to heat during processing, or the use of antimicrobial agents and preservatives that can weaken the cells. Extreme osmotic pressures, or pH levels, can also cause this kind of injury. So, to accurately assess the presence of viable organisms, we need to resuscitate these injured cells. There are a couple of common methods for this. One way is to expose the cells to a simple liquid medium, like a salt solution, at room temperature for about 2 hours. This helps the cells recover from injury. Another approach is to use a solid repair medium, which generally requires a longer incubation time, about 4 to 6 hours. This method provides a more supportive environment for the injured cells to recover and grow so we can detect them accurately. Understanding and applying these resuscitation techniques is crucial in microbiology testing because it ensures we are not underestimating the number of viable organisms present, especially after stress conditions. This step helps us get a more accurate picture of microbial contamination and safety. Now, let's talk about international standards and pharmacopoeial reference substances. These reference materials, including certified reference materials, are essential tools in our laboratories. They help us qualify, verify, and calibrate our equipment to ensure accuracy and consistency. It's important that we use these materials in appropriate matrices to get meaningful results. International standards and pharmacopoeial reference substances serve several critical functions. For example, they help us determine the potency or content of a substance, which is fundamental for quality assurance. They also play a key role in validating our analytical methods, making sure those methods are reliable and reproducible. Another important point is that these standards allow us to compare different methods effectively, ensuring that our testing procedures align with recognized benchmarks. We also use them to perform positive controls, which confirm that our methods are working as intended, and to conduct growth promotion tests, especially in microbiology labs, to verify that our culture media and testing conditions support microbial growth. Overall, these standards are vital for maintaining the integrity and accuracy of our microbiological testing, and they tie directly into the broader theme of ensuring the quality, safety, and efficacy of pharmaceutical products. Now, let's move on to slide 11, which covers positive controls in the context of the growth promotion test within microbiology laboratories, Part II. Positive controls are a fundamental part of our testing process because they help us ensure that our methods are working correctly. When we include a positive control, we're essentially using a sample that we know should produce a positive result. This confirms that our media, incubation conditions, and detection methods are all functioning properly. If the positive control works as expected, but the test sample does not, we can be more confident that any negative result is genuine, not due to a flaw in our testing process. This is crucial for maintaining the reliability and validity of our microbiological testing, especially when we're evaluating products for safety and quality. In summary, positive controls serve as a benchmark, verifying that our testing system is capable of detecting microbial growth when it should, and helping us identify any issues that might compromise our results. When we talk about reference cultures, we're referring to standardized microorganisms that are essential for ensuring the quality and reliability of our testing processes. These reference strains are used to establish what acceptable performance looks like for our media and test kits. They help us validate our methods and confirm that our testing procedures are suitable and consistent over time. Traceability is a critical concept here. It means that the reference strains we use should come from recognized sources, such as national or international collections, or from reputable commercial suppliers. This ensures the strains are well characterized and reliable. Once we obtain these reference strains, we can subculture them once to produce reference stocks. These stocks are then stored properly, either frozen deep or lyophilized, to preserve their integrity. From these stocks, we prepare working cultures that we use routinely in our laboratory testing. Overall, using properly sourced and stored reference cultures helps maintain the accuracy and consistency of microbiological testing, which is vital for ensuring the safety and quality of our products and methods. When working with reference stocks, it's important to remember that if they have been thawed, they should not be refrozen and reused. This helps maintain their integrity and ensures consistent results. Working stocks, on the other hand, generally shouldn't be subcultured repeatedly. Typically, you can subculture them no more than 5 generations or passages from the original reference strain. Of course, this is only if you're following a standard method, or if your laboratory can provide documented evidence that there has been no change in any relevant property of the strain. It's also crucial to note that commercial derivatives of reference strains are only to be used as working cultures. They should not replace the original reference strains, but rather serve as secondary options. All of these points tie into the broader theme of maintaining the quality and consistency of microbial reference materials in microbiology laboratories. Proper handling and documentation are vital to ensuring reliable and reproducible results across different testing procedures. When using reference cultures, it's essential to follow a few key principles to ensure accuracy and reliability. First, all parts of the process should be thoroughly documented. That means keeping detailed records of every stage, from preparation to testing, so we can trace and verify the entire process later. This helps maintain quality and consistency. Second, maintaining complete records isn't just about paperwork. It's about ensuring reproducibility and traceability, which are vital for accreditation and quality assurance. Every step, every decision, should be recorded clearly. Third, performing purity checks and biochemical tests as appropriate is critical. These tests confirm that the reference strain is pure and behaves as expected, which is fundamental for valid comparisons and results. And when we talk about the reference strain itself, it should come from a recognized source, one that's acknowledged by accreditation bodies. This ensures the strain's authenticity and credibility. All of this ties back into the broader goal of maintaining high standards in microbiology laboratories, ensuring our work is accurate, reliable, and compliant with regulatory requirements. Now, let's take a closer look at the reference stocks, G1 through G4. These are typically freeze-dried, stored in liquid nitrogen, or kept deep frozen, depending on the specific requirements. It's crucial to pay attention to the specified storage conditions for each stock because they directly impact the viability and integrity of the samples over time. For example, G1 might have recommended storage times under certain conditions, and those times could differ for G2, G3, and G4. Proper storage ensures the quality of your microbiological samples is maintained, which is essential for accurate and reliable laboratory results. Overall, understanding these storage guidelines helps us manage our reference materials effectively, ensuring consistency and reproducibility in our microbiology work. This is a key part of maintaining high standards in our laboratory practices. Now, let's talk about the working culture in microbiology laboratories, which is a crucial aspect of ensuring quality and safety. First, it's important to understand the specified conditions and recommended storage times for different samples and reagents. These guidelines help us maintain sample integrity and ensure accurate results. Next, routine use plays a significant role in our daily operations. Consistently following established protocols helps prevent contamination and errors, and it keeps our laboratory processes reliable. Since we're focusing on microbiology laboratories in this part, remember that the environment, handling procedures, and storage are all interconnected. Maintaining a disciplined working culture supports the overall quality of our microbiological testing and contributes to reliable diagnostics. So, in summary, adhering to proper storage conditions, respecting recommended storage times, and fostering a routine, disciplined working environment are key to achieving accurate and trustworthy microbiology results. When it comes to sampling, for example, water, it's essential to follow a structured approach. Sampling should be conducted according to a recognized quality assurance system, ensuring consistency and reliability. It's also important to consider any disinfection processes used during sampling. For instance, if sample points are disinfected, this should not compromise the microbial levels within the sample itself. We want to get an accurate picture of the microbial content, so any disinfection steps must be carefully managed. Sampling must be performed by trained personnel who understand the importance of aseptic techniques. They should use sterile equipment to prevent contamination. Additionally, appropriate precautions, like using sterile sealed containers, are crucial to maintain the integrity of the sample from collection to analysis. All these steps are vital in microbiology laboratories especially as we delve deeper into microbiological testing in Part II of this series. Proper sampling is the foundation of reliable results, and maintaining strict procedures ensures that our data truly reflects the microbial quality of the sample source. When collecting samples, it's essential to consider the environmental conditions that might affect their integrity. For example, monitoring air quality or temperature is often necessary to identify potential contamination or temperature-related effects that could compromise the sample. Remember to record the exact time when the sampling takes place. This helps ensure traceability and proper timing for analysis. Proper transport and storage are crucial to maintaining sample integrity. This means using suitable containers and conditions to prevent contamination or degradation. It's also important to monitor storage conditions constantly and keep detailed records of these conditions, so we can verify that samples remained in optimal environments. Assign clear responsibilities for the transport and storage processes. Knowing who is responsible helps prevent mishandling and ensures accountability. Finally, try to test the samples as soon as possible after collection. The sooner we analyze them, the more reliable our results will be, especially in microbiology, where sample viability can decrease rapidly over time. In microbiology laboratories, these steps are vital to ensure the accuracy and reliability of our testing results, supporting overall quality and safety standards. Now let's focus on the key aspects of handling and identifying samples. First, it's essential to have a clear SOP, or standard operating procedure, for the delivery and receipt of samples, as well as their identification. This helps ensure consistency and accuracy throughout the process. When a sample arrives, record all relevant information. This includes the date and, if applicable, the time of receipt. Additionally, note the condition of the sample upon arrival, such as whether it was intact, any visible issues, and, when necessary, the temperature it was received at. It's also important to document characteristics of the sampling operation itself, like the sampling date and the specific conditions under which the sampling was performed. This information is vital for traceability and for understanding any variables that might affect the test results. Once samples are received, they should be stored under suitable conditions to prevent any deterioration or contamination. Remember, storing samples properly isn't just good practice. It needs to be validated, meaning the storage conditions must be tested and confirmed to maintain sample integrity. These conditions should be clearly defined, recorded, and consistently applied. This process is fundamental in microbiology laboratories, as proper sample handling and identification directly impact the reliability of our testing and results. By following these protocols, we uphold the quality and safety standards that are central to our overall approach. When handling samples, it's important to remember that packaging and labels can sometimes become sources of contamination. So, we need to be cautious and ensure these are properly managed. Also, when we perform disinfection, especially on the outer container, our goal is to do so without affecting the integrity of the sample itself. We want the sample to stay as close to its original state as possible for accurate testing. Subsampling plays a crucial role here. It's done immediately prior to testing, following either national or international standards or validated in-house methods. This ensures we're working with representative samples that truly reflect the entire batch. Additionally, having a clear SOP for the retention and disposal of samples is essential. It helps us maintain consistency and compliance and make sure contaminated samples are handled properly. If a sample is contaminated, it must be decontaminated before being discarded to prevent any risk of cross-contamination or exposure. All of these points tie back to our overall goal, maintaining sample integrity and safety throughout the microbiological testing process. Now, let's talk about the importance of quality assurance of results and quality control of performance. Essentially, this means implementing internal procedures to make sure our results are reliable and consistent. For example, we handle deviations carefully, use spiked samples to check accuracy, and perform replicate testing to verify reproducibility. Participating in proficiency testing is also key when appropriate, as it allows us to compare our performance against external standards. The main goal here is to ensure that our results are consistent from day to day and meet the required standards of quality. This is especially critical in microbiology laboratories, where accurate and reliable data can directly impact patient outcomes and public health. These quality assurance practices help us maintain confidence in our work and uphold the integrity of our laboratory operations. Let's talk about quality assurance of results and quality control of performance. Ensuring the accuracy and reliability of our laboratory results is crucial. This involves internal quality assurance or quality control measures, such as handling deviations effectively, using spiked samples to check our methods, performing replicate testing to verify consistency, and participating in proficiency testing when appropriate. These steps help us maintain the quality of our work. The main goal here is to ensure that our results are consistent from day to day and that they conform to the required standards. By doing this, we uphold the integrity of our microbiology laboratories and ensure that our findings are dependable, which ultimately supports better decision-making and patient care. Let's talk about testing procedures. When we conduct testing, it should be based on the guidelines outlined in the national, regional, and international pharmacopoeias. These documents provide standardized methods to ensure consistency and reliability. However, there is room for flexibility. If we have alternative testing procedures, they can be used as long as they are properly validated and demonstrate that they are equivalent to the official methods. This ensures that any modifications still meet the same quality and safety standards. Now, shifting focus to microbiology laboratories, which are a critical part of our testing framework, especially in Part II. It's important to understand that microbiology testing plays a vital role in ensuring the safety and integrity of pharmaceutical products. Overall, adhering to these validated procedures, whether official or alternative, helps maintain high-quality standards across the board. Today, we'll continue exploring microbiology laboratories with Part II. We'll focus on some of the essential aspects that make these labs critical in clinical microbiology. First, we'll look at the types of specimens typically processed. These can range from blood and urine to tissue samples, each requiring specific handling and processing techniques. It's important to understand that proper specimen collection and transport are vital for accurate results. Next, we'll discuss the culture methods used to identify microorganisms. These methods include different media and incubation conditions tailored to grow specific bacteria, fungi, or viruses. The goal here is to isolate the pathogen from the specimen so we can accurately identify it. We'll also highlight the significance of antibiotic susceptibility testing. This process helps determine which antibiotics will effectively treat the infection, guiding clinicians toward the best therapeutic options. Finally, we'll touch on some advanced techniques like molecular diagnostics and rapid testing methods. These innovations help us get quicker results, which is crucial for timely treatment decisions. Throughout this part of the presentation, keep in mind how these laboratory processes connect to overall patient care, emphasizing the importance of accuracy, speed, and proper technique in microbiology labs. When reviewing test reports, it's important to understand how the results are communicated. Typically, if the enumeration result is negative, it should be reported as not detected for a defined unit, or alternatively, as less than the detection limit for a defined unit. This helps clearly indicate that the target was not found within the sensitivity of the test. It's crucial to avoid statting the result as 0 for a defined unit, unless there's a specific regulatory requirement to do so. The distinction might seem subtle, but it's significant in accurately conveying the test outcome. For qualitative test results, the standard reporting language is detected or not detected in a specific quantity or volume. Sometimes, it can also be expressed as less than a specified number of organisms for a defined unit, especially when the number exceeds the detection limit of the method used. Understanding these nuances ensures clear communication and proper interpretation of microbiological test results, which is essential as we move forward in this presentation on microbiology laboratories and their methods. Let's talk about test reports. When it comes to raw data, the key point to remember is that the result should not be recorded as 0 for a defined unit unless there's a specific regulatory requirement to do so. This is important because reporting a 0 when it's not required can lead to inaccuracies in data interpretation. Also, when entering data into electronic databases, a reported value of 0 can be used for data entry, calculations, or trend analysis. This helps maintain consistency and ensures that trends are correctly observed over time. As we focus on microbiology laboratories in this part, it's crucial to understand that accurate reporting of results supports reliable microbiological assessments. Overall, clear and precise data entry and test reports underpins the integrity of our microbiology testing processes and ensures compliance with standards. Vamos a repasar los puntos clave y las ideas importantes de esta diapositiva. En primer lugar, es fundamental tener una buena organización y gestión del laboratorio, incluyendo descripciones claras de los puestos de trabajo. Esto ayuda a definir responsabilidades y mantener un flujo eficiente en las operations. En cuanto al personal, es esencial contar con formación adecuada, qualificaciones verificadas y experiencia relevante. Esto garantiza que las tareas se realicen con precisión y en cumplimiento de los estándares de calidad. Pasando a las instalaciones, debemos asegurarnos de que son apropiadas en cuanto a distribución, diseño, acabado y que tengan áreas separadas y clasificadas según su función. Esto minimiza riesgos y evita contaminaciones cruzadas. Respecto al equipo, es importante que sea adecuado para las tareas, que esté calificado y que se realicen calibraciones periódicas para mantener su precisión y fiabilidad. En cuanto a materials, medios y reactivos, debemos prestar atención a los proveedores, asegurando una correcta conservación y almacenamiento. Además, la preparación y el etiquetado deben seguir protocolos estrictos para garantizar la trazabilidad. Por último, la esterilización, los controls y las buenas prácticas de laboratorio, como las pautas de GPT, son fundamentals para asegurar la calidad y seguridad de los resultados. En resumen, todo esto forma parte de una gestión integral que garantiza la calidad y fiabilidad de los laboratorios microbiológicos, y conecta directamente con el tema general de mantener altos estándares en nuestras prácticas. Let's wrap up with a quick summary of the key points we've discussed. First, sterility testing is a critical component in ensuring that our products are free from microbial contamination. It involves careful consideration of facilities, procedures, and methods to maintain a sterile environment. Validation of test methods is essential to confirm that our testing processes are accurate and reliable. This includes proper sampling and testing techniques, as well as monitoring water quality, starting materials, and the environment itself. When performing testing procedures, we rely on recognized standards such as pharmacopoeia or other validated methods to ensure consistency. Record keeping is also vital. It's important to document actual data accurately, making sure everything is traceable and verifiable. All of these elements come together to support the integrity of microbiology laboratories, which play a crucial role in maintaining product safety and compliance. Remember, the overall goal is to ensure our products meet the highest quality standards through rigorous testing and meticulous record keeping. For further support, please contact us. If you have any questions or need additional assistance, don't hesitate to reach out. Thank you for your attention and engagement throughout this presentation. We're here to help you succeed and look forward to supporting you in your next steps.